Hello everyone, this is MMBS111, Introductory Microbiology. This is mini lecture 12.2, where we're going to discuss the second and third lines of defense. This section is an overview of those general structures, and then we'll get into more detail in section 12.3. There are 5 learning outcomes for this mini lecture. First, be able to define immunology. 2, define marker and discuss its importance in the second and third lines of defense. 3, define PAMPs and PRRs. 4, know the different organs and systems that make up the immune system. And 5, describe the pathway for a stem cell to become a macrophage. All right, so in the next couple of chapters, we're really going to be getting into immunology and the immune system and how our body tries to fight and prevent infections. So immunology is really the study of the body's second and third lines of defense. It's studying these specific chemicals, cells, and processes. It's also a study of the body's response to infectious agents and the study of allergies and cancer. So we're not just looking at bacterial infections or microbial infections, but the study of immunology also includes these other areas. The immune system is responsible for 3 main things. First, it must patrol the body, so it's responsible for body surveillance. It must recognize any foreign material that enters the body. And then once recognized, it must destroy any structures or particles that seem to be foreign, that are not self. The immune system evaluates cells in the body by examining molecules on cell surfaces, and these molecules are called markers. And there are certain markers that we find on our own cells that identify those cells as self. And then microbes also carry their own markers. And these markers help our immune system to recognize them as not part of our body, as foreign. And these are called PAMPs. They are pathogen-associated molecular patterns, and they are different for each microbial organism, but they are markers that our white blood cells are able to recognize that are carried by microbes and not by our cells. Pattern recognition receptors, or PRRs, are receptor molecules on the surface of white blood cells to recognize and distinguish between self and non-self. So when these cells recognize non-self marker proteins, they do not induce an immune reaction. Our cells recognize those as our own and don't initiate an attack on that cell. The immune system is very fluid. It doesn't exist as a single structure or in a single location. It includes a large, complex, diffuse network of cells and fluids that permeate every organ and every tissue. And the structure of the immune system really helps to promote the surveillance of the entire body. So white blood cells are able to move throughout the body and throughout every tissue. And this is also important so that those cells then are able to recognize any particles or structures that are harmful. They are really important in screening tissues, especially blood, for any harmful organisms that have happened to enter the body. There are a number of compartments the text calls them, organs that are involved in the immune system that communicate between those structures. Excuse me. This is known as the mononuclear phagocyte system. This system of sort of fibers and fluids are in direct contact with tissue cells as well as extracellular fluid. Blood vessels and capillaries also penetrate these structures that are included in the immune system that allows these immune cells to travel between structures as well as able to patrol the blood. It also allows cells and chemicals that originate in the mononuclear phagocytic system and the extracellular fluid to diffuse into the blood and into the lymphatic cells and tissues. So the mononuclear phagocyte system, or MPS, is composed of connective tissue fibers. These are reticular fibers, and these fibers permeate the tissues of the body. Phagocytic cells are then sort of incorporated or enmeshed in those fibers and are able to move in the fibers between cells. It's found in the thymus, the lymph nodes, tonsils, spleen, and the lymphoid tissue of the gut and respiratory tract. So this image is also from your text. This sort of shows you the lymphatic system. So the lymphatic system is composed of a network of lymphatic vessels as well as some particular specialized organs. The lymphatic system is designed to provide a route for this extracellular fluid to return to the circulatory system. So eventually this extracellular fluid that is found surrounding cells will be pumped through muscular contraction in the appendages and extremities back into lymph vessels, and then those lymph vessels eventually drain into the circulatory system. The lymphatic system also helps to drain those extra fluids and cells from inflammatory response. And they also provide surveillance, recognition, and protection against foreign materials throughout the body. So it allows lymphocytes, phagocytes, and antibodies to travel through this system. So now we're going to talk about each of these accessory structures to the lymphatic system. Lymph nodes are one of the primary structures. They are small encapsulated or structures. They kind of look like kidney beans, and they filter lymph fluid and provide immune cells. There are a number of locations in the body where you find large aggregations of lymph nodes, and that's in the armpit. These are the axillary nodes. You also find the inguinal nodes in the groin, and then the cervical nodes are in the neck. Generally, when you see your physician, they're going to palpate the cervical nodes to check for swelling. Enlargement of the lymph nodes can be a sign of infection. So your physician is going to check those to see if they're swollen, is there an infection in those nodes. And the next immune organ is the thymus, and it is found in the lower neck region above the aortic arch of the heart. The thymus cells develop specificity. and they are released into circulation as mature T cells. T cells then migrate to the lymph nodes and the spleen, and they remain there to sort of patrol these tissues, and especially to screen blood. T cells are cells that are involved in both the second and third lines of defense, and we'll talk more about T cells in section 3. The spleen is another lymphatic organ. It's found in the upper left portion of the abdominal cavity. It serves as a filter for blood. So it's not really filtering lymph, although lymph fluid is going to flow in these different structures. It's really filtering blood. It primarily removes worn out red blood cells. It also filters pathogens from the blood for phagocytosis by macrophages. So the spleen is composed of 2 tissues, the red pulp and the white pulp. The white pulp is composed primarily of white blood cells, macrophages, that are acting as sort of filters for this blood as it enters the spleen, looking specifically for any pathogens that have traveled to the spleen through the blood. The tonsils. Tonsils house active lymphocytes. They also remove microbes and other foreign materials that enter the body in the food or in the air that we breathe. It was common practice to remove the tonsils prior to our really understanding their function and contributions to the immune system and immune response. Now, tonsils are only removed if there is repeated infection of the tonsils. The final tissue is gut-associated lymphoid tissue, also called GALT. These are small bundles of lymphocytes on or beneath the intestinal mucosa, and they provide this sort of first strike against the influx of microbes that travel through the digestive system. There's a constant flow of microorganisms through the gut, and this tissue helps to address that. The appendix is part of this gut-associated lymphoid tissue. It harbors white blood cells, and it protects the good gut microbiota. For a long time, scientists really didn't understand the function of the appendix, but through the Human Microbiome Project and additional research into the microbiota of the gut, we're slowly learning the function of the appendix and its role in the immune system. The lacteals are lymphatic vessels that are found within the intestinal villi. So the small intestine is composed of long finger-like projections called villi, and each of those villi includes a lacteal or a lymphatic vessel. And then the final component of GALT are the Peyer's patches, and these are aggregations of lymphocytes in the ileum of the small intestine. This diagram is from your text. This illustrates the development of each of the white blood cells as they mature from stem cells in bone marrow. So you can see at the top of the diagram, we have a hematopoietic stem cell. Stem cells are found in bone marrow, and this is where most red and white blood cells mature. However, some of these white blood cells are going to travel to lymph structures, structures of the lymphatic system, and that's where they will mature and become trained to be specific or recognize specific antigens on microbes. So you can see the hematopoietic stem cell leads to two different stem cells, either a lymphoid stem cell. Lymphoid stem cells mature into B and T cells. All other white blood cells and natural killer cells, which are related to T cells, all other white blood cells develop from a different stem cell type. So you're going to have erythroblasts. These mature into red blood cells. Megakaryoblast mature into platelets. Myeloblasts are going to mature into our granulocytes. These are white blood cells that include granules. Those granules contain different chemicals that are released by these cells or are used by these cells to destroy microorganisms. Each of these white blood cells has some certain targets. And then our monoblasts are going to mature into our monocytes, macrophages, dendritic cells. Okay, so make sure you're familiar with all of the types of white blood cells. What's their function? Are they phagocytic? Where do they mature? Do they mature in bone marrow or someplace else? So go through this diagram, this table in your text, and become familiar with these cell types, their functions, and their locations. That's the end of mini lecture 12.2. Here are our checking in questions. Make sure you go through these and be certain that you can answer them correctly. Also remember to go back to the beginning, review the learning outcomes. Be certain that you have acquired that understanding. And here's our critical thinking question. One responsibility of the immune system is to distinguish self tissues and cells from non-self. During an organ transplant, tissues must be tested for compatibility with the patient's tissues. However, it is still necessary to prescribe an immune suppressant after surgery. Why is this necessary? So think about the immune cells and the immune system. How do our white blood cells distinguish between self and non-self? And then why would an immune suppressant be necessary after a transplant? Once you have constructed what you think is a good response, you can find the answer on the next slide. And hopefully this video was helpful.